they have a couple of open reading frames. They encode capsid proteins in this open reading frame called cap, and on the left are proteins involved in DNA replication. So again, this is a small genome, so it can't encode a lot of the replication machinery. Plus, it's a weird structure, so how does it work? The structure is funny because the ends have these terminal repeats which form T structures. So how does that happen? Well, let's look at the sequence at the bottom. This is the sequence of the very left end of the viral genome. And you can see there's the very 3' end, which would correspond to where I've written ORI in the upper left. And you can see the sequences are complementary, but they also form a T at the left end because of the way the complementarity goes. So that's called the terminal repeat, and there's one at either end. And that's going to be the primer for DNA synthesis. Remember, for SV40, the primer was RNA. It's not RNA for this virus. It's simply the ends which are folded back on each other. Now, you're probably thinking, if you use the end as a primer, you're going to lose some sequence, and that's true. So you have to do something special. So we're going to see how that works. So in these viruses, again, replication is continuous. It's not semi-discontinuous or discontinuous. It's continuous. Don't need end. There's no end problem. There's no polymerase alpha because it doesn't have to make RNA primers. That's the polymerase used to make those. It just requires polymerase delta. And the virus encodes 2 proteins called Rep78 and 68. These are needed for initiation and resolution of the products, as you will see. They are endonucleases and helicases, and they bind the 5' end. There is no replication fork. This is done by strand displacement. So let's see how this happens. So the genome is shown here at second from the top with the mostly single-stranded with the 2 terminal T structures. This can be elongated at the 3' hydroxyl at the left end. And the next step, labeled elongate step 2, you see the synthesis of a new red strand using the 3' end as a primer. So that's a perfectly good primer. If we look back at this T structure at the end, you have a base paired segment of DNA and a free 3' hydroxyl. Looks great as a primer for DNA replication. So that's what the virus does. Now, this is done by cellular polymerases, right? The virus does not do this because it doesn't encode a polymerase. So now we have this kind of funny molecule in step 2 where we've made it mostly double-stranded. There's still a T structure at the left end, but that's not good enough to make a genome because we've already lost some sequence at the left end. So what has to be done is this double-stranded product is nicked by the Rep78, 68 protein, which has endonuclease activity. Endonuclease meaning you can cut inside a DNA sequence, not exo. Exo means you cut at the ends. Endo, you can cut inside. So this sequence gets nicked between the A and the D sequences. And then that nick allows the blue part, A', BA, to extend. The base pairing is removed probably by helicases. And so that brings us to step 4, where the blue part is now elongated, and you can fill in the single-stranded region by the polymerase binding at D, the 3' end there, and it makes the red strand. So now you have a fully double-stranded representation of the genome. This is now a complete double-stranded region. So restoring this end required nicking and extending that little single-stranded end. Next step that happens, number 5, is hairpin formation. The left end can form hairpins, and the bottom one looks very much like the initial genome. It's got a 3' end at the end of that T structure, so it serves as a primer for DNA synthesis. The top strand, which is mostly newly synthesized, you can see it in red, comes off. And now we make a fully double-stranded molecule, blue and light red or pink. And the left end has a T structure, which is very much like how we started out at the top here in step 2. So this molecule at the bottom goes around and cycles back into this series of synthetic events. And now we have a genome DNA right here in step 7. So that blue and red molecule is basically a genome DNA. The left end can fold into a T structure, and it could be packaged. So we have, this is displacement synthesis using the DNA of the viral genome itself as a primer. There's no replication fork. there's no end problem, right? Because of this clever business, by using the 3' T as a primer and then nicking the product and filling in the very end, we're not losing any end sequence. And in fact, the sequences at these ends are inverted terminal repeats. And so that's why copying this single-stranded region in step 4 allows you to regenerate the end, so you don't lose any sequence. So that is solution number 2, the single-stranded genomes. How do they replicate? They use themselves as a primer and have a clever way